Cast is now starting. All attendees are in listen-only mode.
Well, good afternoon. My name is Jeff Smith. I'm Vice President of Public Policy, and you are tuning into, I believe, the fourth, possibly the fifth webinar in the AMIA COVID-19 webinar series, where we're highlighting the work of the informatics community during this global pandemic. On today's agenda, we will talk a little bit about AMIA for those of you who are unfamiliar, and a little bit about NCATS and CD2H and CTSA. And then I will turn things over to our experts to talk about a new collaborative that they are launching relative to COVID-19. And then we'll spend a little bit of time at the bottom of the hour, at the top of the hour, listening or having some Q&A with the audience. So to that end, I would ask that you go ahead and put questions into the questions box in your control panel, and we will try to get to as many of those as possible. For those of you who are unfamiliar, AMIA is the professional home for health informatics professionals. Health informatics is the science of how to use data, information, and knowledge to improve human health, including the execution of scientific research, the delivery of healthcare services, and the promotion of public health. AMIA is the multidisciplinary interprofessional home for more than 5,400 such experts. Here is a listing of some of our working groups across kind of the 5 main domains. We have members who advance health and wellness by implementing and evaluating informatics interventions, innovations, and public policy across settings and patient populations, adding to our collective understanding of health in the 21st century through peer-reviewed journals and scientific, increasingly virtually scientific meetings. So before we get into this, because I know we've got several members from outside of the AMIA community, all of these webinars are being made publicly available, and their recordings and slides will be made publicly available. One way to think about informatics is through scales. So we tend to think about patients as an N of 1 or 10 to the 0 power. Practices are usually measured in panels of 1,000. Populations are in the millions, and globally we're talking billions of patients. On the other end of the scale, we have thousands of diseases, millions of small molecules, and billions of lines of DNA. When we apply the scope of health informatics to this scale, it helps articulate the various overlapping boundaries of the 5 main areas of informatics. Translational bioinformatics professionals use informatics tools and methods that link biological entities, like genes and proteins and small molecules, to clinical entities, like diseases, symptoms, and drugs, or vice versa. Clinical research professionals leverage IT and data systems to manage information related to clinical trials, and also involves informatics related to secondary research, use of clinical data. Clinical informatics experts focus primarily on information systems that impact patient care and manage information related to their disease and symptoms, and they do so at the practice level. Public health informatics is the application of informatics in areas of public health, including surveillance, reporting, and health promotion. Public health informatics and its corollary, population informatics, are concerned with groups rather than individuals. And then we have consumer informatics, who are professionals that analyze consumers' needs and information, studies and implements methods for making information accessible to consumers, and models and integrates consumers' preferences into health information systems. Finally, we can think about policies on this scale. And perhaps the policy response to the coronavirus best exemplifies why the globe of health informatics is so important. Across scales, from genomic to disease to patient and consumer, practices, populations, and the entire globe, informatics and the AMIA members have a role to play. So with that as background, I'd like to just quickly introduce, for those of you who didn't know, that this is part of a series of webinars that we're putting on, highlighting how our members are and the broader informatics community are addressing the global pandemic. We've already held several webinars, and we have several additional webinars in the works. And tomorrow, for example, we'll be highlighting the work of some of our nursing informatics colleagues at noon Eastern. And then we've established a kind of landing page of sorts for amia.org/COVID-19, which will be a one-stop shop to find registration details for future webinars, recordings and slides for past webinars, and additional resources that we put together. Okay. All right, so without further ado, I'd like to introduce today's panelists. These include Melissa Handel, who is the director for the Center for Digital to Health, or CD2H, and is at Oregon and Health and Science University. She'll be joined by colleagues Christopher Shute, who is the CD2H co-program director and the Bloomberg Distinguished Professor and Chief Research Information Officer at Johns Hopkins University. And he will be joined by two individuals working, federal partners, working on the federal agency side of things, both Dr. Ken Gersing, who's Director of Informatics at NCATS, and Mitch Aroka, who is the Senior Medical Informatician in the Office of Translational Sciences in the Center for Drug Evaluation Research at the FDA. So with that, I'd like to go ahead and give the virtual microphone over to Melissa.
Thanks very much, Jeff. Hopefully you all can hear me now.
Yep.
Yes, great. Thank you so much. That was a great introduction on AMIA, and we are so grateful to be here today to partner with AMIA to have this discussion about this initiative that the Center for Data to Health and the NCATS NIH Institute are helping to coordinate. The introduction that you gave about AMIA's spectrum of activities is exactly exemplified in why we need to create this national collaborative infrastructure that we're going to be talking about today. And so I hope all of you on the phone can think about how you would best leverage an informatics capacity or in your clinical capacity, a collaborative cohort that's created by all of us for all of us. So with that, I'll turn to some of the many urgent needs, only a few of which are mentioned here, but specifically in the context of AMIA, we simply need better machine learning algorithms and other types of algorithmic approaches to do things like perform rapid diagnosis, triage, build predictive analytics, and a variety of other very necessary features that are very difficult for our caregivers right now. Similarly, best practices for resource allocation, how to best manage hospitals in this time of great need, and then supporting our informatics colleagues in delivering that the information coming from the clinic to our informatics colleagues for drug discovery purposes and being able to leverage information coming from a wide variety of sources collectively. Being able to overall use these types of informatics approaches to reducing disease severity, saving lives, and hopefully, and we're all here today to do this, is to coordinate our efforts to maximize the efficiency and the collective resources that we can all bring together. So all of these things we believe require the creation of a national comprehensive clinical data set that can help us achieve these goals. Next slide, please. So what is the National COVID Cohort Collaborative, or the N3C? So the first thing it is it's a centralized, secure portal for hosting row-level COVID-19 clinical data and deploying and evaluating a variety of methods and tools for clinicians, researchers, and healthcare writ large. That centralized secure portal is secure not only for accessing by, for example, clinicians wanting to ask specific questions, but also for informaticians deploying new algorithms for their evaluation. It's a partnership model. This is something that is being built by a very large number of people as rapidly as possible. But in particular, it's a partnership among several HHS agencies, the CTSA network, there's the distributed clinical data networks such as the CoreNet, Odyssey, ACT-ICD2, and Trinetics, and a variety of clinical partners and informatics experts. This work has been rapidly organized into 4 work streams, and we're going to partition the rest of our panel a little bit around how these work streams are functioning. These work streams are open to anyone who wishes to participate. There are boots on the ground who are being funded to do the work, but given the rapid nature, we need to get things moving along quite quickly. However, we welcome input and contributions from anyone. The 4 work streams are data partnership and governance, phenotype and data acquisition, data ingestion and harmonization, and collaborative analytics. And we'll go through each of what these functions are and how they'll collectively realize the goals of the N3C. The next slide, please. So I just wanted to take a few moments, and we'll come back to this later during Ken's portion of the panel, just to talk about some of the advantages that we see in all the excellent work that's going on in the community, in particular, the advantages of the distributed networks who have already made rapid progress on COVID analytics. And in those contexts, and I'm thinking of CoreNet, ACT, Trinetics, and OMOP, we have the ability to maximize the number of records. We can already query very large numbers of records, not only nationwide, but worldwide in some cases. We have a lot of flexibility in the diversity of querying, and we have more complete longitudinal data. Now, these approaches are really great for launching, immediate implementations, and there has been a lot of work in all 4 of those networks to help support COVID analytics. And so the questions that a user may have are sent to network partners where those data reside locally in that clinical data model. And then the aggregate answers are sent back to that user. So I wanted to contrast the advantages of the distributed networks with what's on the next slide, please. So here, a centralized harmonized COVID-19 data set has a different set of advantages. So here, we aim to create a large data set for the N3C. We will provide harmonization across all the different clinical data models and data sources that we're able to in the short amount of time that we aim to do this. There will be the ability to launch the types of machine learning applications and analytics that really require patient-level, row-level, comprehensive EHR data for patients diagnosed with COVID, possible diagnoses with COVID, or negative control. And finally, the approach here can be purpose-driven curation and data modeling specifically to take advantage of what we can gather from the EHR relating to COVID-19 patients. Next slide, please. So I'm going to just briefly go over the data partnership and governance work stream. This is led by John Wilbanks at Sage Bionetworks. And just to kind of reassure you, it looks like we've had some issues in our translation into PowerPoint. The clinical institutions over there on the left sit behind agreements that are formed. So there's a central IRB. This is being hosted at Johns Hopkins in based upon the earlier work that Johns Hopkins did in establishing the central IRB to the All of Us program. There's also a central data use agreement that institutions would sign with the NIH in order to support the data ingest into this cloud environment where we would have our harmonized COVID data. Qualified researchers down there on the lower left, clinicians and data contributors who may or may not be a member of one of those clinical institutional partners, but would have to request access via a data access committee, which has stakeholder representation from the various institutional partners, agencies, and clinical data model sharing networks. Out of that clinical COVID data there in the center, we would have synthetic derivation to create an open data set that would be freely available to any researcher anywhere with a simple registration and access process so that we can track usage and make sure that people, according to the data use agreement, give the knowledge back to the community if they find anything useful in the use of those data. Next slide, please. So I just wanted to quickly go over the data use agreement principles. So since the data could be identifiable to the patient and institution, as we are keeping geocodes and timestamps, because those are both very important for COVID analytics in particular, these analyses are only for the analysis of COVID, e.g. Community spread, risk and treatment. There can be no re-identification of patients or contacting of patients. It is only used for research, public health, and development activities relating to COVID-19. There will be a limited data set, which is de-identified as much as possible, but as I mentioned, in this case, there is a risk for re-identification, so we have it as a limited data set. The platform is FedRAMP secure, and we'll talk a little bit more about that later. And as I mentioned, we'll have the data access committee approval process. The requirements are that those using the data would have to abide by these terms of agreement. There would be a time period for the use of agreement. We're still discussing what that time period would be with the community, the valid IRB that would include these limits, and that any findings would be shared back to the consortium, as I mentioned. And finally, there would be no secondary redistribution of these data. These data are not allowed to leave that secure enclave that is hosted by NCATS. Next slide, please. I'm going to hand it off to Dr. Shute to talk a little bit about the next two work streams here, the phenotype and data acquisition work stream.
Thank you very much, Melissa. And as you can see, this is really two work streams that we're going to talk about. The first one is acquisition to establish a common phenotype, enable services for sites to send their data to us, and of course to ingest the data into a secure location. Next slide. I should add that this is being led by Emily Pfaff, who's our community liaison in the CD2H Next Generation Data Workstream. And she's created, in collaboration with many of the clinical data model communities and people working in this area, a phenotype, or essentially a proto-query that is a consensus drawn from across many networks that would describe what kind of cases we're going to request from the CTSA sites and other contributors, such as the VA. And this is outlined here with specifics for what constitutes a lab-confirmed positive case, a lab-confirmed negative person, which has implications because eventually, potentially everybody would be lab negative confirmed, and we don't want the entire EHR downloaded. This is not a replication of everybody's EHR into a common data set. Likely positive categories, and then possible positives. As you can see, the scope of data to be pulled is essentially what exists in the common data models: observations, specimens, visits, procedures, and so on, in a way that would be potentially useful to data discovery and exploration in a COVID data environment. Next slide. This is a sort of a sketch of the entire workflow from soup to nuts. You can see that consensus phenotype that we just described on the lower left, feeding into the explosion of a single site hub environment where it's a dealer's choice to use the local common data model, be it PCORnet, Trinetics, OMOP, or ACT, or a hybrid with their local data warehouse. Regardless, we are hoping they would send the data in a common data model that would then be staged, harmonized, and put into a production database. We're going to expand a little on that. Next slide, please. This is historical work that's been done, and I should add that CD2H has been busily doing what we call plumbing for the past 2.5 years, anticipating this precisely this kind of opportunity to generate a cohesive, generalized data set across the CTSAs. We didn't anticipate COVID, of course, but we've been doing a lot in partnership with the federated query community with a data model harmonization. And this is an example of the work that had been done at mostly NCATS in partnership with CD2H, using the Adeptia tool and creating models from one version to another and linking them to a common framework. All of that data was stored in the CADSR environment, which will create a great boon for informing us about model-to-model transformation as we move forward in this project. Next slide. One of the first things to happen is, as they extract their data, they would choose which CDM to use. We want one and only one CDM. We don't want duplicate copies of the data from the same site. They would take a query of that proto-phenotype that I showed you, which will be pre-written in each common data model syntax and language, create a local folder, and then transfer that to NCATS. We'll have substantial support for the sites to undertake this. Help desk, subject matter experts. Each of the common data model communities have been quite forthcoming in bringing subject matter experts to the table that we can consult with to ensure that this is done as accurately and as completely as possible. They'll execute the CDM code, make COVID data augmentations, which are future things. We know that, for example, some codes, not everybody codes the LOINC tests yet for COVID fully in their organization, so there may be workarounds for that, and then transfer the data. Next slide. When it's extracted, we're going to have first of many stage quality checks. We'll reincarnate the data they send to us as flat tables into the clinical data model of instance that they chose to send it to us. So we'll have hopefully 60 odd of these reincarnated clinical data models against which we will use the common data model tooling native to that environment. So if it's the CoreNet data instance, then we will use CoreNet tools and so on. That when we discover issues or variants, and in particular, the use of data elements or value sets that might include expired or deprecated terms. I confess Johns Hopkins was guilty of this for a while. We had in our RX norm codes that had been deprecated or made obsolete. That level of check will happen at this phase to ensure that we have as complete and as comprehensive and as accurate a data set as possible in the data model that they sent to us. Next slide. We then have moving into the harmonization work stream, where we'll take the data that we've hopefully captured from each site in the secure staging area and mapping it into a common framework. The common framework will, of course, be OMOP 5.3. Next slide. We'll use the Adeptia tool, which is a commercial ETL tool that NCATS has purchased and has substantial experience with, where we take that reincarnated, hopefully reasonably cleaned CDM instance, and then using the ETL processes of Adeptia, informed by the maps that we have created and updated through previous work, generate an instance in OMOP 5.3 of the same data that had been contributed by the hub. We'll identify variations from the local instance as that second data quality check. So if things are systematically missing in OMOP 5.3, then it's probably a transform error that we'll double check at this phase. Next slide. We finally have the third data quality check, where once we have the data in an OMOP 5.3 instance, we'll leverage the fairly rich and robust tooling of the Odyssey community to do a data quality machinery dashboards and quality checks. We'll return those dashboards to the contributing hub sites, but we probably won't ask them to act on it at that phase. We hopefully can address issues discovered at this point in subsequent refresh cycles. Next slide. Then we conduct the major merge, which is this flotilla of OMOP databases, each one of which has been, to the extent practical, quality checked, validated, and complete, will be merged into a combined hub data instance of the merged OMOP 5.3 instance that will be migrated to the analytic platform. Next slide. Just a word about future. While our tactical emphasis for the next 6 to 8 weeks will clearly be wholly and exclusively on OMOP 5.3, we do expect over time to achieve an aspiration where we can create FHIR as this pluripotent intermediary in partnership with CTSA hubs and members. Not that it has to be a centralized repository, but we can create data that could then be transformed algorithmically into say, STDM files for submission to FDA, as well as generating algorithmically derivatives into the common data models that are widely used. Next slide. So with this, I'm going to introduce Mitra Roca, who's going to describe much of the work that has been done previously in the Common Data Model project that informs the current process. Mitra?
Okay, thank you, Chris. Next slide. So as Chris mentioned, I'm going to talk about the Common Data Harmonization Phase 1 accomplishment and Phase 2. So this project has been funded by HHS ASPE, Assistant Secretary for Planning and Evaluation, and have the investment opportunity called Patient-Centered Outcome Research Trust Fund. We are one of the many projects that has been funded under this portfolio. Next slide, please. So the goal of this project is to build a data infrastructure for conducting research using real-world data from administrative claims, as well as electronic health record data. And for this project, our objective is to develop a method to harmonize several common data models from 4 different networks, allowing researchers to be able to ask questions from a larger pool of data, as well as leveraging open standards and control terminology. And this project is a collaboration between FDA and 3 institutes from NIH, NCATS, NCI, NLM, as well as from HHS ONC, the Office of National Coordinator for Health Information Technology. Next slide, please. So here I try to take the work we are doing and comparing with different countries using different outlets. And what you see in the white is the intermediary model that we use to harmonize these 4 common data models: Sentinel, ITB2-ACT, BCORNet, and OMOP, to an Uber model or intermediary model. And for Phase 1, we use the bridge model, Biomedical Research Integrated Domain Group. Next slide, please. So the proposed solution is that the FDA reviewer, EPICO researchers, other researchers from NIH or academic medical centers will have access to the portal and the tools that have been developed by NCATS. And through this infrastructure, they will have access to data in any of these 4 common data models: Sentinel, OMOP, BCORNet, and ITB2-ACT. And then, as Chris mentioned, we use the Adeptia ETL tool to be able to generate CDISC standard, CDISC STTM, as well as HL7 FHIR standard. Next slide, please. So these are some of the objectives for Phase 1 of the Common Data Model Harmonization. So we developed the general frameworks, tool processes, governance, and standards for transformation of these 4 common data models, and we collaborated with NLM on maintenance and sustainability, as well as NCI. Then HHS ASPE asked us, they didn't want this to be an informatics exercise, but to be able to answer a real clinical question. So we collaborated closely with the oncologists from FDA, as well as oncologists from the National Cancer Institute, and they developed a protocol focusing on immuno-oncology products in patients with autoimmune disorders. These patients are excluded from clinical trials, so we wanted to see if in real world these drugs are prescribed to patients by healthcare providers. And then one of our other tasks, number 3, was to be able to reuse the infrastructure that has already been funded by ASPE PICO Trust Fund projects. And 2 of these initiatives are the NIH CDE repository, as well as Data Access Framework, which is a HL7 standard. Next slide, please. And the other 2 goals of these projects are to leverage open standards and control terminology to advance patient-centered outcome research. And then the main goal is to be able to test methods and tools that have developed by one collaborative on this universal CDM mapping and also transformation approach. Next slide, please. So Phase 1, actually has been completed. We are just finalizing the final report, which will be submitted to HHS. So in phase 1, we have harmonized 5 common data models, Sentinel 2 versions of PICONET 3.1 and 4.0, OMA 5.2, and IDB2 Act version 1.4. And we harmonized them with the bridge, this intermediary model. And then the infrastructure, which Dr. Ken Gersing will talk about that in detail, has been stood up at NIH NCATS, and that will enable us to query, view, and store the results. And then we have been using open consensus-based standards. In phase 1, we collaborated with several data partners with Mayo Clinic and Yale. They are part of FDA's CRC program, Center of Excellence for Regulatory Research and Innovation, as well as collaborating with Eligo Health Research. And through Eligo Health Research, we collaborated with 4 different data partners: IQVIA, Hospital Corporation of America, Flatiron, and University of Chicago. And the goal was to collaborate with them on executing the query on their data, and they could select which common data model they would like to use. And then in parallel, I collaborated with Harvard Pilgrim Healthcare to develop the Sentinel query for our oncology use case. Next slide, please. So phase 2 of Common Data Model Harmonization Project has been funded by 21st Century Cures Act, and it started on October 15, 2019. We are actually identifying new data partners to collaborate with us on this new use case that we are focusing on. And our goal is, if the data partners would like to use any of the common data models, they can do that, or if they would like us to query the electronic health records directly or their clinical data repository using HL7 Fire, we are actually working on that infrastructure at NCATS. So we are going to use one of the HL7 Fire standards to standardize the queries, and the standard is CQL, Clinical Quality Language, and then also being able to, as also Chris mentioned, be able to generate the CDISC STTM, which is one of the standards that is required by FDA CDER and CBER for submission of clinical trial data to FDA, and then taking it through the FDA gateway and do the validation. Now I'm going to hand it over to Dr. Ken Gersing, who is going to talk about the architecture.
Thank you, Mitra. I appreciate everybody being here and taking the time to listen to us. So my piece of the presentation is about really the last phase, which is the analytics. You know, we spend a lot of time talking about common data model harmonization and making everything work together, but actually in some ways, even though I'm very much a believer that that's the foundation of all science, then we really need to make it usable so the scientists can take it from the people, from the informaticians who've made the plumbing all work together and discover new things to help people get better. So that's what I'm going to talk about today. Next slide, please. So this piece of the project's headed up by Justin Ganay at Sage Bioworks and Peter Robinson at Jack's Lab. But I just wanted to go back to a little bit of what Melissa was talking about, federated versus a central model, and to see what is that really delivering, and how do they specialize in kind of different questions. And so I put these two next to each other. There's really so in a federated model, you would ask the typical question: what is drug X? How is it beneficial to a COVID patient? Or does disease Y impair the course of COVID? Or does an income greater than $50,000 a year improve a person's outcome? Those are very specific questions. But in a centralized repository, what it's allowing us to do is ask questions that are much more open-ended. So what drug or drugs help COVID patients? What different diagnoses impact patients? So you could imagine having diabetes or hypertension, as we've heard about. Obviously, lung disease, smokers may impact your outcomes. So instead of saying which one of these, you can just say, using tools like machine learning, say, let's find correlations with these. Or the social determinants of health might be, instead of, what is your income, like we asked on these more pointed federated question, we could say what social determinants of health are correlated with improved outcomes or not improved outcomes. So that's what we're really pursuing. So next slide, please. So the other thing that Melissa mentioned, next slide, please. There we go. The other thing that Melissa mentioned is that this is a limited data set where we're keeping dates of service and a geocode. But there was a second thing that Melissa mentioned, which was the ability to produce a completely synthetic data set or a computer-derived synthetic data set. So this is actually a sub-project it's not the main path, but we wanted to talk about it because one of the tremendous disincentives to do a centralized data set is getting it by the regulatory patient protective regulations that are appropriate but make it just hard to get the data together. So if we could actually produce a truly synthetic data set that was representative of an EHR, then a lot of those barriers would be would come down and make it much easier. And I'm presenting a slide that was from Philip Payne at WashU, where they took a tool and they trained the tool to look for sepsis prediction. And then they took the synthetic data set and they compared it, the machine modeling of random forests, and you can see how they compared. And if you look at the final two columns, you'll see that how it performed on the real patient data on the left of the two columns, and on the right column, you can see how closely it compared. And so we're pretty excited about testing this out. If this works, then instead of the limited data set, there will be a truly synthetic data set. And it's like Melissa said, just by registering, you would be able to use this, bring it down actually, unlike the enclave data set, and play with it locally. Next slide, please. There you go. So everybody has talked a little bit about FHIR and phase 1, and I can't help but putting in my, excuse the pun, plug towards that also. I think that there's a lot of, this project is going to continue that work, even though we're focused on COVID, because we're still pulling a set of this and placing it in the FHIR repository. And the idea with this is bulk FHIR coming to it maturity, that we will be able to combine these data sets to be able to ask a single question. And just to keep in mind, the idea is that we should be able to not only harmonize it to ask questions, but then export it in the format that's appropriate for the HHS agency or investigator of choice. And so our hope is that we're producing a common infrastructure across not only NIH, but we're working with CDC and FDA, ONC, NLM to try to say, look, if we use these common standards, that we should be able to share this, and then we can export it as CDISC, SCTM for FDA, or we can export it as a JSON file for CDC or NIH if they want a more traditional CSV. So just one slide forward. There you go. So that standard that we keep talking about is actually the slide, and I promise not to go into too many details on it. But one of the things that you're seeing here is that with the common data models in the bottom left, sorry, the blue part on the left are data partners. Think the University of X, and they are bringing in their data into the model of choice, as Chris is doing. But in the FHIR project, instead of bringing it into an OMOP aggregation, we're actually aggregating that from a FHIR repository and then going through much of the same process. So on the right ends up being HHS agencies, but the output is as they need for their specific requirements. Next slide. There we go. So one of the things that we're talking about is once the Chris's team has harmonized the data and the ingestion, and we're getting it in an analytical model. In this case, it's OMOP 5.3, but again, in the future, hopefully, a FHIR representation. It's got to go into a secure platform for analytics. And there are a lot of really wonderful platforms out there, but because there's such an emergency and we need to get going quickly, we have access to a analytics platform called Palantir, which we're using. This platform is actually shared at the White House, CDC, FDA, and many NIH agencies, including NCATS and NCI. We actually share, at NIH, we share the same platform with NCI. It's actually the same instance of it. And the reason is that it's FedRAMP ready right now, and we don't really have time to have other applications make that available. And so in the future, if the community said, look, we really don't want to stick with Palantir, I just wanted to emphasize, we understand that, and this is not trying to lock folks in. I just need to emphasize that it's what we have available now in the immediate need. Having said that, Palantir is actually a very nice program. Amazing amount of security. The origin of this company is security and banking. So, you know, everything that you would imagine. It's FedRAMP certified. It's, you know, granular. You have access. You can role-based access to the cell. There's ongoing 24/7 monitoring of the applications, but looking for you know, any security breaches. It's got security audits throughout the program. One thing also really nice about Palantir is it is an ecosystem. It does have its own tools, but we, in our instance, we have Tableau and with SAS, and we have R. So you can bring your own tools. You can even build your own tools in Palantir, and it's quite accommodating to that. Data can come in and out of the system, and it can also handle multiple data types from PHI, clinical, diagnostic, genomic, and imaging data. And it can handle time-sensitive, time services data type, which is quite nice. I talked about it's open with other tools. It supports open API. It comes with a machine learning and supports NLP. It has a complete version history, and which will help with reproducibility. That's quite nice, and versioning of the different data sets as they come in. And then a couple of features, I already mentioned a few of them. You can bring your own tools. I listed here SQL, Python. It has a complete lineage of the provenance of the data sets. And as I mentioned, you can bring in lots of different tools. So the idea is that when this synthetic data set, when this limited data set goes into the Palantir instance, any investigator, like Melissa said, that goes through the data access committee or the central IRB, depending on their level of security needs or their level of investigation, will have access to this program to use this tool. Bring your own tools, bring OMOP tools, bring any tools you want, and investigate the data. And this will be both intra and extramural, meaning folks inside the NIH or inside HHS or outside of HHS work for the community. Next slide, please. One of the things I want to talk about is, if we bring all this information, why would the community help? And obviously, in this case, there's an emergency and where there's a health reason to participate. But the other thing is that we know that the informatics community often works on things and does not get credit for them. And in the reality is we understand that the way you get a raise or promotion is, you know, publish or perish, obviously. And so we're putting an enormous amount of emphasis on making sure that people who contribute to this effort, whether it's an artifact or contributing to work, or actually an organization that contributes to the data, or any of the above, gets credit for these through, you know, with the persistent identifier on the work they've done. So this can be used for them to show that the tremendous donation of work that they've done and effort they've done. Next slide, please. So this is a work in progress. We are going as quickly as we can. I can't tell you that this won't change because of everything that's evolving. But at this point, this hour, this minute, Obviously, NCATS is moving forward with supporting it, and CD2H is coordinating this. We have been working very closely with NIADS and NLM, National Heart, Lung, and Blood, and the Cancer Institute. The Cancer NCI folks have already started posting images. They're in charge of the imaging and shared imaging side of Palantir. And so some of those are actually already on the system and will be outside and live in probably days. And then, as Melissa talked about, the data model networks, the common data models, have been very generous with their time and support, and we very much appreciate PCORI and Active Odyssey and the Trinetics team. And then the folks participating right now are FDA, the VA, CDC, and DoD are in discussions to work with us on donating data and helping us with the project. And next slide, please. Melissa, this is just how to get involved. I don't know, Melissa, if you want to take this over, but there's many ways to get involved. And these are some of the ways that you can get involved with the work that the CD2H is coordinating. Thanks
so much, Ken. Yeah, I'll just say a couple words. So there's a bit.ly there at the top. First, again, I just want to thank everybody who has been participating and contributing their resources, their time, their vision to this initiative. As Ken mentioned, it's really exciting to see the community come together and be able to leverage our existing work, all the work that's been going on collaboratively in doing the clinical data model harmonization and the terminology services and the cloud architecture, as well as all the foundational work that the different institutions and clinical data model data sharing communities have had ongoing. And so this is really about putting all of our pieces end to end and working together to try to create this cohort for everyone's use. So we would really, really welcome everyone's participation and your ideas. And so the onboarding information is there, and we look forward to seeing you on the meetings and in the Slack. So thanks very much to everybody, and we'll take some questions and discussions.
Great, thanks, Melissa. And for those of you who have not provided questions in the questions box on your control panel, though you can go ahead and put your questions in there. We already have a fairly good amount of questions, so thank you in advance for all of those. And let's see here. Okay, so I think a couple of things, a couple of softball issues that I think we can start off with, and then certainly I'll try and group several questions as we get the opportunity. We've got about 10 minutes here, so I do think that there's potentially a need to just kind of clarify in terms of timeline. Do you have a sense of timeline when you anticipate data becoming available to conduct analyses? And from your perspective, are there specific governance pieces that still need to be put into place?
So I'll take the first crack at that, and then I'll hand it off to Chris. So we anticipate having test data in this sort of, base structure that's been instantiated this week. And then we're hoping that the platform will be functional for any users to use within the 4 to 6 week range. And we recognize that this, of course, is not fast enough for everyone's needs, but in order to have it be robust enough to garner the kinds of insights that we know we need, we feel that's a good target. As the groups are still really defining all the particulars, and of course will depend upon the ability to get data according to the regulatory needs of the institution, which could also, of course, slow things down. However, we feel pretty confident that because we've based a lot of the work on the IRB and the data use agreement on existing work and partnerships, that hopefully that burden wouldn't be too high. But I think Chris can give an update on the central IRB and the processes for that.
Sure, and I do want to give a call out to Sage Bionetworks, who've done an outstanding job in helping us draft the data use agreement. It's been a collaborative effort. Many eyes have been on it. Correspondingly, for those of you that on board, you can join one of the work streams and receive the information and content. One of them is the governance and effort, where just this weekend we sent out the draft of the full protocol for the IRB. And the community is literally, I'm staring at the Google Docs page as I speak, and see quite a crowd there contributing to our protocol and identifying issues and questions and issues of refinement. We hope for the central IRB process to be completed early next week, in an effort to really make it easier for collaborating sites to participate and not have to burden themselves with their own IRB. However, the central IRB is not mandatory. If a contributing site chooses to go through their own IRB, that is quite acceptable. There's no prohibition to it. It's just we're trying to minimize the amount of work involved.
Great, thanks. And just to confirm, Chris, Johns Hopkins is the central IRB in this case?
That's correct.
Okay. All right, thanks. Okay, a couple technical questions. I think this is not unexpected. Can you speak to the quality of FHIR instances of patient data seen thus far, if you've seen any FHIR instances in the wild? For example, consistency of the representation beyond basic codable concepts like dosing frequency and timing and meds. So can you speak to the quality of data FHIR instances that you've seen so far?
Yeah, I was almost afraid to bring it up. I knew that was a bit of a red herring. Let me reemphasize that our tactical plans over the next 6 weeks are to focus exclusively on OMOP 5.3 as the target transformation model. When we talk about FHIR, and I did certainly, we see that as a strategic issue. I agree that the state of FHIR maturity today is, how do we phrase this, variable. There are some FHIR resources that are extremely mature, most are not, and as is the state with the FHIR APIs into EHRs. Some people that have recently updated their EHR versions do have FHIR Force services, but not the full suite of services that the ONC has stipulated in its recent regulations. That is a work in progress. That's fully recognized. I think it's a fair statement to say that FHIR is not going to be a major contributor to the N3C initiative in the short term, and conceivably not until very, very late in the process.
Okay, yeah, thanks for that. All right, so here's a question on contributing data, and this is really around a couple process pieces in terms of, can you elaborate on the frequency of data feed updates that can be expected as part of this? And then a kind of similar or adjacent question is on, for site contributors, do they have to agree to be part of this synthetic derivative in order to participate?
That's a good question. Oh, no, go ahead, Ken, you answer.
Yeah, let's see, no on the synthetic. The synthetic is definitely, there are two kind of parallel experimental paths going on. The synthetic is definitely one of them, and the other one is FHIR. And we didn't want to, I hope we didn't talk too much about that, because I want to just emphasize it right now. This is a COVID crisis, and OMOP 5.3 is the analytic set, and will be, so no, you do not have to. As a matter of fact, in phase 1, only 3 institutions have identified to do that, and we've already identified those. Regarding the frequency, I think that I personally believe the biggest delay in this will be getting the institutions to sign the data use agreement, and that kind of that human factor, but also getting NIH to feel comfortable, because they are actually becoming a co-signer, which is somewhat new for them. NCATS is completely okay with that. My boss, Chris Austin, is going with that, but we still very much want to get this cleared because we think this is an important precedent for the NIH in general. So we're working on it this morning, and your answer to your question is we're going to negotiate with the folks, but ideally we would like it something like every 48 hours for 3 months, and then as hopefully the crisis slows down, we would get it less often every week for a couple months, or and then every quarter, whatever month, and then every quarter. We haven't decided that, but we know that most sites will not be able to do that at that frequency, but that is what ideally we would like.
Okay, a couple more questions here on participation. Can you state again how many healthcare delivery organizations have committed to providing actual patient data? And then can you confirm that to participate, a site must be able to host their data using one of the identified clinical data models?
Right now, we're only right now we're only taking those 4 to answer that part of the question. I defer, Melissa, if you want to speak to the other half of the question.
So as Ken mentioned, we can't really accept data that isn't in one of those 4 clinical data models. That's really what we have the bandwidth to help support. There is, for each of the clinical data models, going to be a small group of people that we're calling the white glove service that will help a clinical institution get their data into one of the target models so that we can ingest it. And so in that case, for some institutions that don't already have a model, if they're close enough, we could support helping them get their data into one of those 4 models. As far as how many institutions, we have quite a number of CTSA institutions involved in this process. We also have, as Ken mentioned, organizations such as the VA who are going to be pushing their data as well. And then we also have some additional clinical institutions that are not currently CTSAs, but are also interested in pushing data and also find some of the systems. There's some typing going on. Sorry, everybody.
Okay, thanks for that, Melissa. In terms, one more participation question: Does a specific institution need to contribute data for researchers from that institution to be able to get access to the data repository, or does a researcher who wants access to the data repository have to be from academia?
They do not need to be from academia, but they need to abide by the data use agreement requirements, which include sharing the results of the knowledge gained back to the public. So if you're a commercial informatician and you want to participate in the use of these data, the data cannot be removed. They cannot be used for selling anything, and the knowledge must be contributed back. But we really want to inspire, all informaticians everywhere to help contribute to the COVID initiative. So we're trying to design the DUA and the DAC process to support that goal. So we don't want to close off informatics people who come from industry.
Great, thanks. Probably just a couple more questions here. Can you talk about the coverage of the data geographically? Will this be representative of COVID-19 patients who are test positive but not hospitalized? And will this include patient outcomes later for nationwide serological testing data?
Well, I can address the serological data. I mean, we are asking for all laboratory data to be given, and when serological data becomes more widespread, I know it is technically available, but it's hardly widespread. Presumably it will have its own LOINC code. It may already, I'm embarrassed to say, I don't know. But we would have that kind of outcome data. And obviously with an update refresh rate that may decelerate over time, we would, by virtue of that design, capture outcomes as downstream sequelae of COVID infection or non-infection as a comparison. I'll stop.
Okay. Well, unfortunately, we are at time. We've got a lot of really interesting questions here. So what we'll try and do is we'll convene the group, and as a function of updating and making this recording available on the AMIA website, we will try and answer some of these other questions. I just want to take a moment to thank the presenters and to thank all of the attendees in Internet land for taking the time out of their busy schedules to learn about this. I think we can all agree that this is a tremendous opportunity to leverage a lot of investment that the NIH and others have made, including the FDA, and really a lot within the informatics community. So I will say good luck to the presenters as they proceed with this work. And I will remind everyone else that we will have additional webinars as soon as tomorrow, and then additional webinars will be scheduled over the next few weeks, focused on telehealth and telemedicine, doing a deep dive there. And then we're going to be having a pair of webinars focused on how the public health informatics community is engaging on COVID-19, as well as other quadrants of AMIA. So thank you all very much for the presentation and for the great questions. Good luck, and we'll see you next time. Thanks, everyone.
Thank you.
Thanks so much. Bye-bye, everybody.